Okay, hello. Thank you for joining us and welcome to El Zoominario. Today I'm very excited to introduce Horacio Lopez-Nicora. Um, actually, I should have asked you, is Nicora or Nicora?
Nicora.
Nicora, okay. Horacio Lopez-Nicora. He's originally from Asunción, Paraguay, earned his bachelor's in agronomy from Universidad Nacional de Asunción and a master's in crop sciences from the University of Illinois Urbana-Champaign, and both a master of applied statistics and a Ph.D. in plant pathology from Ohio State. He's currently an assistant professor at the Ohio State University, focusing on improving soybean health through plant pathology and nematology. Before joining Ohio State in 2021, he held various academic positions in South America, and he continues to serve as an adjunct professor in Colombia and Paraguay. Thank you so much, Horacio, for accepting the invitation, and then you can take it from here.
Okay, excellent. Thank you, Claudia, and I'm very happy to be here. So yeah, like Claudia mentioned, my name is Horacio Lopez-Nicora. I'm an assistant professor at Ohio State. And I was born indeed in Paraguay, and I did my bachelor's in agronomy. And I'm gonna walk you very quickly here through my academic journey. But I'm providing this QR code, and I'm going to provide several resources through QR codes. This one here will contain a list of other works that I'm doing. I'm gonna only be able to share two of them today, but if you're interested in talking more about other works, please check that out and send me an email. So I did my master's in crop science in University of Illinois Urbana-Champaign, from where later I went to Ohio State to get my Ph.D. in plant pathology and a master's in applied statistics. I returned to South America for 3 years. I worked in Universidad San Carlos in Paraguay, where I founded the first plant clinic in Paraguay. Plant clinic is a hub, a diagnostic lab that essentially can take any problem a grower have, like field crop, vegetable, ornamental grower, any issue that they have with their plant, they can bring it to a place where that problem can be diagnosed. And that type of lab did not exist in Paraguay. And we founded the first one while I was there. And I'm still working with the research team in that university as an adjunct professor. I'm also an adjunct professor in Universidad de Caldas in Manizales, where we do a lot of research in banana, plantain, and now soybean. There's a foliar nematode that we're now studying and trying to better understand the etiology and epidemiology. But at Ohio State, I am the soybean pathologist and nematologist, so I focus on diseases that affect soybean. Um, my research program really focused and tried to understand the multiple interaction between different pathogens that can affect this crop. And you can replace soybean. Soybean is my favorite crop, but you can put here your favorite plant, and there will always be multiple threat to that plant. And Howard Samuel Fawcett said very well in his presidential speech. He was the president of the American Phytopathological Society, and he told us that nature does not work with pure culture. So we do have multiple pathogens that can interact with each other, causing more or less disease. And that's really what I'm interested in learning more. So I'm building my research program on top of these three main pillars: pathogen detection and surveillance. So trying to always stay one step ahead of the epidemic outbreak, disease outbreak. Pathogen host interaction and resistance. So how can a pathogen overcome resistance? Or how can a pathogen develop resistance to some of the chemical products we use to control them, some of the fungicide? We're doing research to understand that. And also understand the role that the plant pathogen plays in the agroecosystem. And you can see that conducting research and multiple experiments trying to answer these questions will lead to a lot of data that then we have to analyze and very accurately produce results. Because my position has a three-way split, I am 40% research, 40 extension, and 20 teaching. I do respond and I try to develop research that answer relevant questions that soybean growers have in Ohio. But I definitely will not be able to do all this without the amazing team that I'm building, and I'm acknowledging them right now: grad students, postdoc, undergrads, and lab managers that help me conduct research in the lab, in the greenhouse, and in the field. I'm very proud of the team that I'm working with at Ohio State. So as the soybean pathology, I have to handle any threat that we're exposed to during the season. So it can be from multiple species of Fusarium, some of them associated with sudden death syndrome that we have to deal and better understand, to white mold, fungal pathogen that causes fluffy growth, mycelia, and can really be very severe, producing disease in soybean. You can see an affected soybean completely destroyed here next to one that was not affected, that clearly can produce viruses that can cause necrosis, foliar pathogen, other true fungi that can chew and destroy the root system of our plant. Fungal-like organisms like oomycetes, such as Phytophthora and Pythium, can also cause severe disease in soybean. And then last year, we have an outbreak of seed and pod decay caused by multiple fungi. So we're exposed to different threats, and we have to every season come up with experiments that collect all this data to better understand and develop management strategies. I'm very passionate about teaching. I teach field crop diseases and apply nematology in autumn and spring semester, but I co-teach Code Club with Jessica and Yelmer, and we do this every semester. I'm providing the QR code there to direct you to our website where we provide all the tutorials. This is really a club. We teach every once a week, and we teach our programming. So not statistic per se, but mainly how to analyze data, how to wrangle data, how to plot and visualize data. And we have been very successful with this class. So I invite you to visit some of the sessions that we put out there. Okay, so with that, and I want to focus the conversation today, focusing on two pathogens that affect soybean. And we're going to use these two pathogens to talk about some experiments and how we use this data-driven approach to manage disease. One is the soybean cyst nematode. It's an animal, and the other one is Macrophomina fasciolina. It's a fungus. They're both pathogens of soybean. They're both soil-borne pathogens. They lived in the soil. They can overwinter, survive for a long period of time. The nematode as eggs, and the fungus produces a survival structure called microsclerotia, and that's the way that it can survive. So let's start with the soybean cyst nematode, and the first project I want to share with you. The soybean cyst nematode is arguably the most economically damaging pathogen of soybean. It can cause significant yield reduction, sometimes even without visible symptoms. So we can have plants growing out there that look healthy, yet they're losing a lot of yield. They can overwinter as eggs. They will hatch as the nematode, penetrate the root, and then the female can lay on average 250 eggs. And this can happen 4 to 6 times during a field season. So you can see how rapidly the numbers of this nematode can explode in one season. The SCN Coalition, which I'm a co-leader of, recommends the following for managing soybean cyst nematode. Know your numbers. So we need to test our field, collect soil samples, and test for soybean cyst nematode. Rotate sources of resistance and soybean variety. Rotate to a non-host, a host that will not allow the nematode to reproduce, and consider a seed protectant in some cases. Again, the QR code will take you to the SCN Coalition website. You can see all the projects that we're working on, all the information in terms of understanding better the biology of this pathogen and management strategy. So I invite you to visit the SCN Coalition. So since sampling a field is the most important and proactive way of managing soybean cyst nematode, and the way we do it is manually, With Richard Gonzalez, a master's student that graduated last year in my lab, we wanted to try something else. We wanted to try an automated sampling to see if we were getting very similar result as we got with our manual sampling, which we will consider as our gold standard. So when we have to sample large areas, doing it by hand takes a lot of work. As a matter of fact, for his project, we were in the field between 5 to 7 people for 8 hours sampling each of the points, whereas with the automated sampling, we were able to do it in less than 2 hours with only 2 people involved in this operation. So how does this automated sampling work? It's a big knife that you can see will collect soil. It will collect soil and pull that soil sample, and with an air compressor, it will deliver the soil, and at the same time will clean that knife that collects the soil sample so there's no cross-contamination. The soil will be placed in this plastic container, and this plastic container will have a QR code that will be linked to the GPS. So every single soil sample that is retrieved will have a GPS coordinate. We will collect and process these samples to know the numbers of eggs of soybeans in nematode per 100 cubic centimeters of soil, 100 cc of soil. And because we have the information from every single point we collected sample, we can generate these type of maps. We can use an inverse distant way interpolation technique to predict where or how much nematodes we have in places that we did not sample from places that we actually collected samples and quantified the nematode. And what you can see here, hopefully, is that the map that is generated with the samples collected with the automated samples are very similar to the ones that we generated when we collected samples by hand. And we did it in different fields that had different soil textures and soil conditions. So we're seeing that the maps agree pretty well. We're also seeing, and we quantify, that there is spatial aggregation. So in areas that are very red, where we have a lot of nematodes, those areas tend to aggregate in space. So we have cluster aggregation of the pathogen in a field. We want to assess how well these two techniques work, and we use a concordance analysis. So we have our gold standard here, the manual sampling versus the automated sampling, a 45 degrees line. And if the accuracy or if the concordance or agreement was perfect, all the points would have fallen in that red line. However, we see that it doesn't happen that way. But we're not accounting for that spatial aggregation here. If we grab those different levels of soybeans nematode that we generated with our interpolation map, and we can actually go and capture those pixels from each of those little quadrants and use that to calculate that concordance analysis, and then we average for each of those categories, we now have an almost perfect agreement between our manual and automated sampling technique, which is exactly what we see when we compare those maps side by side, right? And this is great because it has a lot of practical applications. For example, a grower with such a map will now be able to decide to only rotate to a different crop in some part of the fields that are more heavily infested with the pathogen, rather than planting a different crop all across the field, including in areas where there's very little pathogen. A scientist can also decide where to put some trials in that field to be able to assess either different seed treatments or cultivars, and that's exactly what we did with the growers. We were able, with this map, to decide where to put different trials to answer questions that the grower have. Does the seed treatment work reducing the nematodes population? What does using a different cultivar does in terms of yield increase? And we were able to do this because we know that putting the experiment in this area, we actually had the pathogen and we could test this. Moreover, we can get yield maps very similar to these and overlay to maps that contain information about pathogen. So very clearly, we demonstrated here that our automated sampling was working as an alternative to the manual sampling, a very time-consuming, you needed more resources, and we can now cover very large areas in very reduced amount of time. We were also able to integrate these with geospatial tools to more quickly identify the spatial distribution of the nematode, and with that information, include on-farm trials. So with that example of how our data-driven approach could help us identify localized areas of threats in a field, I wanna now to jump and share a different study, and I'm gonna take you now to Paraguay. This is a work that we just recently published and got a press release. So if you hit that QR code, it will take you to the press release for this publication. And we're gonna see how we're using data analysis to understand the wealth of information that we've gathered from this project. Dr. Sandeep Mondal, a postdoc in my lab at that time, worked with me on performing many of these analyses. So we went to Paraguay in this area. This is the southern part of Paraguay where we do a lot of the agriculture. A lot of the soybean is planted in this area. And we went and sampled more than 300 soybean fields. So we're covering different departments in Paraguay where soybean is produced. And we collected soil samples. We visited those fields when the soybeans were planted. So we definitely knew that we were collecting soil samples from soybean field. We also identified that there were areas affected by Macrophomina fasciolina that causes charcoal rot in soybeans. So we can see some premature dying of plants. And when we went to those areas and actually pulled the plants, we could see those microsclerotia actually on the stems and root. So the plants were dying of charcoal rot, and we could actually see the pathogen present. When we compare at the large scales, we saw that the different departments have different and significantly different levels of the fungus per gram of soil. So we quantify them as colony forming unit. So we can quantify this, and we saw that in some areas like Itapúa, where soybean is produced a lot, we had more abundance of Macrophomina fasciolina. We also started exploring the aggregation at a larger scale. So in the previous example with soybean cyst nematode, I was showing the spatial aggregation at a field level. Here we apply a different type of geospatial statistic to evaluate that aggregation, but at a larger scale. We're now essentially looking at almost like a whole country level, right, with the data that we collected. And we used other techniques like hotspot analysis. From those soil samples, not only we quantify the pathogen, but we also send it to the soil lab to get all these different edaphic factors or variables: pH of the soil, organic matter, texture in terms of percentage of clay, and other variables that we can use. And what I'm trying to show here is that all these variables are highly correlated with each other. So when we have this type of data set, it just yields out to maybe use a principal component analysis to reduce the variability and reduce the numbers of variables. And that's what we did. We reduced everything to 2 principal components, which explained a lot of the variability in that data set. And so now what we wanted to do was to use some of geospatial statistic tools to model and create maps that will inform areas of risk for abundance of this pathogen. And you can see that the first one that we use is the same one that I presented before, IDW, inverse distant weight. But this one is just interpolating. It's using the information from the points where we sample to try and give an estimate of how much of the pathogen is present from places when we did not sample. But there are so much more information in our data set that can be used. So then we try a kriging approach, which not only does the same type of interpolation, but it gives you a variance. And not only that, it also allows you to test different models. So we're evaluating which model fits best our data set. And furthermore, what we did is we use a co-kriging, and the co in this case stands for covariate. So we were able to incorporate all the information from those edaphic factors that I just showed, that we put it in two principal components, and we're able to fit into the modeling of this map, improving even better to tell us where we were able to have more or less pathogen depending on what area we were going to. We were also able to test different types of trees and different models to tell us, for example, in areas where we had more heavy soil, we were going to find more Macrophomina fasciolina, or pH being one of the very important and significant variables. We were able to demonstrate that in areas that we had more acidic soil, there was a higher risk of having Macrophomina fasciolina. So these spatial random forest models that we evaluated really show us some of these variables, that normally you get with your soil analysis that could be used as a predictor for Macrophomina in different areas. Including this spatial dependence of the edaphic factor into our different models really improve our maps. And with this, of course, we're helping growers and policymakers to inform themselves with more data-driven information and decision of how to manage charcoal rot in these areas of soybean production. And with that, of course, I want to thank funding sources and the team that helps me. And without them, I really couldn't be here presenting most of these research. So I will entertain some questions, but before that, as part of the Zoominario, Claudia asked me to present some cultural traditions from Paraguay. And I had a very long list, but I wanted to highlight these three ones and maybe a couple others if I have more time. Paraguay is really one of the few countries that is bilingual. And when I mean bilingual means that the law is written in both Spanish and Guarani. And Guarani is the Aboriginal indigenous language of that region. So we still spoke that language. And we preserve the Guarani myths and oral traditions and cultural practice, especially in rural areas of Paraguay. It's fascinating. The tereré is a ritual. We drink a lot of mate, yerba mate. It's very common in Argentina, Uruguay, Brazil, Paraguay to drink mate, which is a hot beverage that you pour on yerba mate. But tereré specifically is pretty much the same, but we do it with cold water. So we normally have a cold water, and we normally put some herbs such as mint to give some flavor and make it very refreshing. And we drink this, and we normally tend to share this with friends. As a matter of fact, when someone comes to your house, you will offer a mate, or you will offer a tereré in this case. And then the Paraguayan harp is one of my favorite instruments, and it's used a lot in our country's folk music tradition. So used in genres such as the Paraguayan polka and the guarania. I strongly recommend you go and find some guarania music. It's just lovely. You're gonna really appreciate it. And then, of course, I need to present some type of food, right? When we talk about tradition, this is the sopa paraguaya, so the Paraguayan soup, very confusing for many who goes to a restaurant in Paraguay and wants to order a soup, and then a cornbread comes to the table, right? So that's the sopa paraguaya, very traditional. And then empanadas, probably empanadas is very common in Paraguay, but the very traditional thing in Paraguay is to buy a empanada and a bread and make your empanada sandwich. So you will have your empanada sandwich. That's very typical in Paraguay. So with that, I'll stick around for any questions. Thank you.